Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Welcome back to the deep dive. Today, we're tackling something that gets, I think, far too little attention in the world of high-performance biotech.
It really does.
We spend so much time on the magical parts, the amazing strains, the media recipes. But one of the biggest reasons those projects fail at scale is because the concrete and steel couldn't keep up.
That's absolutely right. We're deep diving into facility engineering. For too long, people have treated the manufacturing plant as just passive infrastructure, a container.
Just a box to put the tank in.
Exactly. But in microbial fermentation, the facility is often this silent, unyielding, limiting factor. A strain that's a total rock star in the lab can have its performance just completely throttled by the utilities at 10,000 liters.
And that is the core mission today. We are unpacking why the building and its utility systems are not passive. They're an active performance system that controls reliability, sterility, and, ultimately the bug's output.
Okay, let's unpack this then, because this shift is crucial. In the lab, you have incredible flexibility. If you need more oxygen, you just crank the valve. The supply is basically infinite for your needs. If the temperature drifts, you nudge it back.
That person fixes it.
A person fixes it. But at manufacturing scale, the facility becomes what we call deterministic.
Meaning what exactly?
Meaning the utility systems, the steam, the compressed air, the cooling water, they set hard, unavoidable boundaries. You are physically limited by the compressor size or the chiller capacity or just the diameter of a pipe.
Okay.
So if those systems are poorly designed, they define the entire operating window for everything. They constrain the actual performance the organism can deliver.
I think that lab versus plant contrast is so key for anyone listening. They get the biology, but they need to see how the physics of the plant dictates that biology.
Right. In the lab, deviations are fixed by a scientist. In the plant, deviations are, they're structurally imposed limitations.
They're built in.
Precisely. We need reliability, and that means the facility has to deliver repeatable boundary conditions. The organism needs a consistent experience batch after batch. Same oxygen, same substrate, same shear.
And if the facility lets those things fluctuate,
then the cellular experience is inconsistent, and that's where you get batch-to-batch variability, even with the exact same strain and media.
And the practical implication of that is, it's huge. You could spend millions on strain optimization, but you just see diminishing returns.
Because the strain can't express its potential. The plant is literally overriding the bug's genetic blueprint.
Okay, so what are these constraints? Let's break them down.
We can sort of group them into three big areas. First, sterility, boundary, integrity. So your cleaning, your sterilization, your closed piping.
Okay.
Second, gas delivery stability, keeping that oxygen transfer rate, or OTR, steady during peak demand.
And third?
Heat removal capacity. You're dealing with metabolic heat plus the heat from compressing all that air. If any one of those fails, the organism fails. Genetics don't matter at that point.
Okay, let's talk about the physical world then. Let's start with the building itself. Spatial layout and flow control. How does the geometry of a facility prevent contamination?
In microbial plants, it's all about flow segregation. This is more than just clean rooms. It's about rigorously separating the flows of personnel, materials, and waste.
You're creating dedicated highways, in a way.
Exactly. You have to physically separate raw material prep from the sterilized operations and, crucially, from harvest and waste handling.
Because the raw materials for microbes are so rich, right? They're food for everything.
They're perfect food for everything. Complex carbs, nutrient broths. They're a buffet for contaminants. So that interface where waste streams meet raw materials is an incredibly high-risk design zone.
I can imagine.
Your spent media, your cleaning fluids, those have to be completely isolated from, say, your incoming sterile media or your pure inoculum.
So the goal here, the gold standard, is always closed processing.
It is the absolute default. Once we sterilize that vessel, we have to maintain the sterile boundary with robust valves, filters, closed connections. So engineers, they prioritize these closed designs at the points that fail most often.
And that would be the transfer steps.
Yeah. High-frequency transfers, adding the inoculum, sterile feeds, antifoam, and of course the final harvest line.
I can see how just growing a facility over time messes this all up. What happens when you retrofit a pilot plant?
That's the brownfield reality.
Yeah.
And it's where you can permanently engineer in structural failures. You bolt on a new skid and suddenly you've created problems. Bad piping geometry, dead legs, poor slopes that don't drain, complex manifolds, things that become almost impossible to sterilize properly. The building itself becomes the source of contamination.
That makes perfect sense. So the building sets the rules. Let's get back to the dynamic systems, the utilities, because that feels like where the day-to-day variability really lives.
Yeah.
These utilities as active process enablers.
Let's start with SIP sterilization in place. The mindset has to be that this is a complex unit operation, not just a compliance checkbox you tick.
What's the difference there?
It's recognizing the physics of it. When you're steaming media, you're adding energy and volume. You have to manage evaporation, condensation. The facility must be designed with robust condensate management. You need volume reconciliation. If you don't track the water you've lost or gained during steaming, you've already changed your starting media concentration.
Wait, hold on. So before the bug is even in the tank, the process of just cleaning the system is already changing the recipe.
Yep.
That's a built-in facility-driven source of batch variability right there.
It is. And with steam itself, the most common weakness isn't the temperature. It's poor steam distribution and condensate drainage, especially in complex accessories.
Like what? Sampling loops.
Sampling loops, pressure sensors, vent condensers, all these little nooks and crannies.
Yeah.
If you trap air in one of those spots during SIP, it creates a cold spot. That's a guaranteed sterility failure waiting to happen.
Okay, let's switch to oxygen. For any aerobic microbe, breathing is everything. It sounds simple, but I'm guessing it's not.
It's probably the most constrained variable we have. Oxygen just doesn't dissolve well in water. So the facility design has to treat gas header pressure stability as a critical process KPI.
Why the header pressure specifically?
If that pressure dips, even for a moment, it changes the gas flow rate, the shear at the sparger, the whole bubble regime inside the fermenter.
So there's a direct physical link between a pipe in the utility room and the microbe's ability to breathe inside the tank.
A direct link, and here's where it gets really interesting. The antifoam effect.
Ah, yes.
At scale, you have to use antifoam. You can't have foam blowing out of your vessel, but it has this powerful negative hydrodynamic consequence.
What's the hidden trade-off?
Antifoam, usually silicone-based, changes the surface tension. This stops big foam bubbles, but it also stabilizes the thin liquid films around the small gas bubble.
And that's bad because
It actually hurts mass transfer. It can reduce your oxygen transfer efficiency by 10, maybe even 25%. So you add antifoam to stop the batch from foaming out.
but you end up starving the bug of oxygen anyway.
Exactly. You trade one very obvious failure mode for a much more subtle one.
That is walking a tightrope. So you're sacrificing efficiency for containment. Now let's add viscosity and heat to the mix. High cell density means thick broth and lots of heat.
And when microbial broths get really viscous, it totally changes the oxygen landscape. In tall vessels, you get these intense vertical OTR gradients, meaning the dissolved oxygen sensor near the impeller at the bottom might read 20% saturation, while the DO near the top of the vessel is less than 5%.
So the scale itself creates zones of oxygen starvation. The microbes are having completely different experiences depending on where they are in the tank.
They are. And this leads to a huge warning about scaling. Utility systems do not scale linearly. You can't just double the pipe size for a doubled vessel. You have to co-design your heat removal, your aeration capacity, and your mixing power. The solution has to be exponential, not additive.
Let's move to that critical pinch point.
Yeah.
USP, DSP equipment iteration. The harvest step. This sounds like where sterility goes to die.
It's the transition zone. The second you start moving that concentrated, nutrient-rich broth, you're dealing with high solids, foam, and constant valve manipulations.
Yeah.
So the facility design must clearly define that boundary shift. Are we staying sterile? Or are we moving to sanitary but not sterile handling? Everything downstream hinges on that one decision.
And I can see how the facility's physical limits could immediately compromise the biology you just spent weeks growing.
That's the hidden cost. DSP often starts with a centrifuge or a filter. If the building, say the floor loading or the ceiling height, forces you to buy an undersized piece of equipment,
That limitation feeds right back upstream.
right, you might have to deliberately lower your target cell density or dilute your broth just so your undersized centrifuge can handle it. The building structure is setting your biomass target.
And if you have to wait for that undersized equipment, you create hold points. That pause is a risk in itself.
A catastrophic risk. Intermediate holds in these rich broths are just begging for contamination. A facility needs engineered policies for them. minimize the time, control the temperature, and ensure they are perfectly cleanable and drainable. A whole tank can't be an afterthought.
Which brings us right to hygienic equipment and piping design. Beyond just having a shiny surface, what's the most critical design element for cleanability?
Oh, it has to be drainability. Any liquid you leave behind, anti-foam, product residue, spent media, it creates a cold spot during SIP, and it seeds biofilms for the next campaign.
Cleanability first, I like that.
It has to be the philosophy.
So we have to talk about the infamous dead leg. We all hear about these 3D, 6D rules, but how should an engineer really think about them?
Dead legs are still a dominant contamination driver, absolutely. They trap liquid during cleaning and block heat during sterilization. But we need to move beyond simple length rules and start to risk rank them.
Based on what they're used for.
Exactly. A dead leg on a sterile addition line or a critical harvest path carries way more risk than one on, say, a utility return line. Those high-risk ones have to be engineered out completely.
So if drainability and geometry are the foundation, then CIP must be the sterility engine of the whole plant.
It is. Microbial plants live and die by their cleaning in place. CIP performance has to be a core facility KPI. And you have to validate it not just with sensors, but with cleaning studies, using real fermentation soils from your process.
Not just some generic challenge.
Right, because your specific product residues might be much tougher to clean than a standard protocol assumes. If we connect this to the bigger picture, remember we said facility failures are rarely these big catastrophic events.
Right, they're subacute.
They are. They erode your yield, they shift your impurity profile, they introduce variability, but they don't cause an immediate alarm. And that makes diagnosing them incredibly hard. It takes long-term data trending and a really deep mechanistic understanding.
Okay, so give us the diagnostic signatures. How does a facility problem actually show up in the process data?
Let's take a classic. High cell density E. coli. That bug is incredibly sensitive to oxygen. If you see gas header pressure transients, a failure that starts in the facility's compressor or header, it causes these momentary OTR deficits.
And what does the E. coli do in response?
It immediately shifts to overflow metabolism, stops making your protein efficiently, and starts pumping out acetate, which is a waste product that inhibits its own growth.
So that little pressure dip in a pipe could cost you what?
It can cut your final protein yield by 15, even 20%. The data makes it look like the organism is failing, but the root cause is the stability of your gas delivery.
But how do you know it's a pressure dip and not, say, a mistake in the media feed rate?
That's the key diagnostic question. A feed error would be gradual. A facility pressure issue causes an immediate coupled response in your sensors. You'll see a sudden, sharp drop in the DO controller output that's completely mismatched with the organism's oxygen uptake trend.
Ah, so the data tells you the delivery system failed, not the consumption system.
You got it. The data tells the story.
What about the viscosity example, something like Bacillus actioniformis?
Right. When that broth gets thick, say 20 centipoise, those vertical OTR gradients we talked about become lethal. The localized oxygen starvation at the top of the vessel triggers a stress response in the bacillus.
And the biological result of that stress.
It induces protease activity. Proteases are enzymes that chew up other proteins, including the valuable enzyme product you're trying to make.
So the plant's physical limits are causing the bug to destroy its own product.
Exactly. That stress response, rooted in vessel geometry and utility limits, can erode your final product activity by 10 to 30%.
And finally, those latent sterility risks. The SIP underperformance. What's the signature of that across a campaign?
It doesn't kill batch number one. It shows up as sporadic post-SIP bioburden positives. Little random hits of contamination that get more frequent as the campaign goes on. It's the classic signature of a biofilm, seeding in a cold spot and slowly growing over time.
So what does this all mean for the listener who's just trying to maximize their biological output?
It means facility engineering is the reliability multiplier. Proactive management of utilities, of hygienic geometry, of architecture. It's not a cost center. It's what's essential to actually unlock the biological expression you've worked so hard for in the lab.
It's the bridge from the lab bench to resilient manufacturing.
It is.
It sounds like the true measure of success isn't just preventing a total batch loss. It's managing all these subacute failures.
Precisely. It's about successfully controlling those OTR deficits, or finding that biofilm source. Because if you don't manage them, these facility-originated problems are guaranteed to disguise themselves as failures of the strain or the media.
That's a really profound takeaway. So the next time you see batch-to-batch variability, don't just blame the bug. Ask yourself, are we measuring the constraints of the plant? or the true potential of the biology? Which one is actually holding the other back?
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.